Okay, well, welcome everybody. My name is Robert Carter. You now know that. I am a speaker for Creation Ministries International. If you're not familiar with CMI, I'd highly encourage you to go to our website, creation.com, when this is all over, because you're going to find a lot of articles on this subject, and a lot of other subjects, a lot of things that I've written, a lot of things that a lot of other people have written right there for you. Tonight's topic is going to be difficult, especially because I'm going to challenge a lot of sacred cows. I'm actually going to challenge some specific places of our biblical understanding and probably some of the assumptions that we're making that are incorrect. But we're going to wade through that. So just prepare to be, stretched a little bit, because what I'm about to show you, I think, is very consistent with the Bible, but it might not be consistent with some of our views of Scripture. Oops, how do I go to my next slide? So creation.com is there for you anytime you like. But also CMI, because we're an information ministry, we have to connect to people. Now, I know a lot of you on this list already get our weekly email newsletter, but if you don't get that, I would highly encourage you to sign up for it. It's really easy. All you have to do is either go to creation.com/infobytes or text your email and zip code to this phone number, 470-344-INFO. 470-344-INFO. Now we need a zip code, because every time we send a guy out on the road, we'll email everyone within a certain area and say, hey, come to our event. And everywhere I'm going, I'm getting people who are coming there because they've gotten a notification like this. But our events are shut down until the end of July. So once we start up again, once all this coronavirus stuff goes away and the world is free once again to proceed as normal, we're going to be starting our speaking ministry. So I really appreciate you giving me this invitation, because without this, our ministry, we would have nothing to do. As we're reaching out and figuring out how to connect with people electronically, this is really cool to be able to do this. All right, I need to establish myself as a geneticist. If you look at my bio, you'd see that my PhD is in coral reef ecology. That doesn't make any sense until you realize that while I was in my coral reef ecology program studying these beautiful animals, I got my degree after I stole the genes out of these animals that code for these bright green and bright red proteins, and I engineered them into these fish. So half of my doctorate was about genetic engineering. And ever since then, that was 2003 when I finished at University of Miami. Ever since then, I've been studying data, writing computer programs, analyzing the human genome, analyzing the 1,000 Genomes Project, analyzing Neanderthal genomes. A huge part of my life has been wrapped up around genetic data. And I'm going to tell you right now that some of this data has been challenging, but almost all of it has been exciting because of what we've been learning that challenges the evolutionary story. In fact, anything you thought about human races that you learned maybe 10 years or more ago is almost certainly wrong, because everything's been flipped upside down. In fact, it's really cool. This might be the only time in history or in some field of science we've had more data than we've had theory. There's so much data, no one single person in the world can get their mind wrapped around everything everyone's learning. That's how fast this stuff's coming at us. All right, you've heard of the Table of Nations, Genesis chapter 10, right? You've heard of the 16 grandsons of Noah, I'm imagining. A lot of people, when they read this, they say this: Ham went to Africa, Japheth went to Europe, Shem went to Asia, or something like that. Most people, depending on where they put Shem and Japheth, most people put Ham in Africa. But this is actually biblically naive. The Bible does not actually say that. You might have seen maps like this, where they take those 16 nations that came from Noah and place them on a map. Okay, this is really interesting. But notice the geographic spread of these points. It doesn't even get into Europe. It doesn't get into Africa beyond Egypt. It doesn't get into Iran, let alone Afghanistan, India, or China. This is a very small portion of the world. So we have a question. Was this written to describe everything after the Table of Nations, or was it several hundred years later someone wrote about all the nations that he was familiar with and what branch of Noah's family tree they came from? Those two very different ideas. This is either description of people as they're spreading out, or after they spread out, someone wrote wrote what he knew about. See, I think it's the latter. But if it's the latter, we're not told where the sub-Saharan Africans came from. We're not told where the Europeans come from. We're not told where the Native Americans come from, the Australian Aboriginals. Most of the world's population is a giant mystery. And that makes this really, really interesting from here on out, because it means that we are free to just take the data that we find and examine it. So what we're going to do, we're going to step backwards. Most of the time, when you hear people trying to describe the people in the world, they start in Africa and they go outwards. Well, I'm actually going to start with the Native Americans. North and South America were one of the last places on the earth to be settled. Actually, New Zealand was probably really one of the last, but I'm not going to talk about Pacific Ocean so much. We're going to start with Native Americans and go backwards to Babel. But we have to use ancient DNA. In fact, if we use modern DNA, we get some curveballs thrown at us really quickly. You know, this is a map. This is a map of the most common Y chromosome in Europe. I carry this Y chromosome. 3 million other Irish men carry the same Y chromosome. And this is where all my Y chromosome relatives are found in the world. But my branch in the Y chromosome tree is actually pretty recent. All these men were very closely related. There are men in the world that are much more different than all the people in my little family of Y chromosomes. Okay? Who are those people down there in Africa? What is that? Well, in Cameroon, south of Lake Chad, in the dead geographic center of Africa, live some of the most darkest skinned people in the world. And they're my Y chromosome brothers. In fact, my Y chromosome might be like 80% of Western Europe, which means 20% of Western Europeans have Y chromosomes on different branches of the family tree. So it's my Irish great-great-grandfather, great-great-great-grandfather, I forget. He comes over from Northern Ireland in the middle of the Irish potato famine. I could be more closely related to one of the darkest skinned men in the world from Africa than my Irish great-great-great-grandfather was to his next door neighbor. What is a race? What an awesome question. In fact, scientifically, we can't define races anymore because we can't find DNA that's exclusive to one race and not others. Let me put it this way. There is not a single letter in the genome where 100% of Africans have that letter and no one else in the world has it. There's not a single letter in the genome where 100% of Europeans, 100% of Chinese, 100% of anyone, everyone has that letter and no one else has it. Whoa, wait a minute. That means you can't use single letters to define a race. Now, there are some letters that are more common in Africa than Europe. Okay, so statistically, you can say this person, if you're looking at only the DNA, this person is more likely to be African or Siberian, but you can't do it perfectly. So the whole race question gets opened up. But before we get into the ancient part of this, we have to give you a timeline of everything. We're trying to take all this information and put it into a specific timeline, a biblical timeline. Well, here I've rounded everything off to the nearest 500 years. So creation is it's a little more than that, but it's about 4000 BC. The flood is about 1600 years later. Abraham would be about 2000 BC. Moses about 1500 BC. David about 1000 BC. Jesus at 0. And today we're at 2000 AD. So that is a very rounded off, rough sketch of biblical history. Now we have to put 7 billion people in this time frame. We have to put Africans and Icelanders in this time frame. Can we do it? In fact, we have to put it in a time frame since the flood. Yikes. There's a lot of genetic diversity in the world. is this just ridiculous? Why would I even bother trying this? Well, I wouldn't bother trying to say this in public unless I didn't think we could do it. I'm trying to set it up to make it sound difficult. There's something we have to talk about is the Bronze Age. Because the Bronze Age is going to be very important when we get to talking about Europe, when the modern Europeans arrived in Europe. Yeah, the Bronze Age is before the time of King David. King David was in Iron Age 2A, I think that's the specific archaeological context. But before the Iron Age was the Bronze Age. Just, you know, the Trojan Wars and all that kind of stuff happening. We call that the Bronze Age. There's Troy right there. Actually, was Iron Age. Interesting, right? Right before King David was born. This is when the Trojan Wars are happening. Peloponnesian Wars between Athens and Sparta were way late in biblical history. We're talking about, you know, the Bible's talking about the great grandsons of King David, who are, you know, kings, whichever king it was. I'm just guessing here. I don't know. But the Peloponnesian Wars are happening about that time frame. All right. Everyone got a picture of the history? Now let's get into ancient DNA first. Ancient DNA is not supposed to exist. It's not supposed to be there. DNA breaks down too quickly. Now, I might expect some DNA, if we dug up the body of Moses, I would expect some highly fragmented DNA to be found, maybe. 15, I say it's 15 plus 2, that's 3,500 years. That's a long time for DNA to decay. And so it was a shock in, I don't have my notes, I think this is the late 90s, I think. Someone went to a museum specimen of a quagga. A quagga looks like that. They're extinct, like a zebra horse sort of thing. They're extinct. This is a museum specimen, a zoo specimen, but they had it in the museum. So he went and he took a sample and they looked to see if there was DNA in it. And lo and behold, DNA had lasted from 1883. That's over 100 years. Everyone was shocked. We can put this in context here. We're now pulling DNA out of things the evolutionists are claiming are 800,000 years old. But 10, 20 years ago, it was a shock to find DNA in something that was 100 years old. Ancient DNA is not supposed to be there chemically. In see, I don't have my notes because we're on a screen share here, but 2000 and something, 2005, I'm going to guess, is around there. A graduate student in Svante Pablo's lab, who's now running a big thing at Harvard, he was asked to pull DNA out of a Neanderthal bone. And he didn't want to because there's no DNA in Neanderthal bones. But he did it, and he did it, and there was DNA in there. And it shocked the world on multiple fronts. First was the presence of DNA. Shocker. Second was the DNA that they found. Because you see, it wasn't monkey DNA. It wasn't half man, half human DNA. It was human. It was so human-like as to be ridiculously human. So you've, back in the past, you've seen all these pictures of Neanderthal man, like this skeleton here, this is in the New York Museum of Natural History. Notice that both of his knees are bent. Why are both of his knees bent? It's because to make him look like he's a monkey when he walks. But from the hips down, he looks like you. In fact, morphologically all over, this is an extremely human-like specimen. Now I know, we've been conditioned to think them as half man or at least primitive at least. But look at that charcoal drawing there. He's got big brow ridges, okay, and he doesn't have much of a forehead, but he made his face stick out to make him look more like a monkey. That's from the 1800s. But look at the lower picture there, the two skulls. The Neanderthal skull's on the right. His brains are bigger than a modern man. On average, Neanderthals had larger brains. They had stronger muscles, thicker bones, thicker tooth enamel. They were very robust people. They were built. We're puny by comparison. They call us gracile, thin. But the Neanderthal was robust. And you know what? They survived in an Ice Age Europe. I don't think I could survive there. I don't think any of us could. These people were tough. But we're now pulling DNA out of horses and cave bears and mammoths and mastodons and all sorts of crazy things from way far in the past, according to the evolutionist. and the DNA is there. Okay. But most ancient specimens don't have DNA. Pick up a random bone from a random grave site in the world, there's probably not going to be any DNA in it. Some do, but what happened was someone realized that the petrous bone, that's the bone that contains the inner ear, the densest bone in the body, can often have over 100 times more DNA than other bones in a grave. And once they realized that, they started pulling the temporal bone and pulling the petrous bone out of that. And this is when the field of ancient DNA exploded, because all of a sudden they had 100 times more samples with DNA than they thought. And today we have thousands upon thousands of ancient DNAs from humans. Now, most of those are from Europe because this is where the technology started. We got a couple from Africa. We have some from North America, South America, some from China, but most of them are from Europe currently. Okay, there are three big reasons why we would not expect ancient DNA to be in an old bone. First is from something called cross-linking. DNA is a sticky molecule, and under oxygen and water and things like that, if you have two DNA molecules touching, they will literally react and form a covalent bond together, and your DNA strand becomes a bird's nest. It becomes, and when that happens, as the molecules are wiggling and stretching, it creates tension points and it fragments. This is actually a photograph of cross-linked DNA from a 4,000-year-old ancient Egyptian mummy. That is really, really cool. But cross-linking is a major problem. Another even bigger problem is called cytosine deamination. Oh, big words. Cytosine, you know the four letters of DNA, A, C, G, and T. Well, C is cytosine. There's a cytosine there on the left, circular molecules, got some double bonds, NH on the bottom, double O, and there's an NH2 on the top. That's an amine. That's what makes urea and ammonia smell. This is what makes proteins have lots of nitrogen in them. Is right there. Sorry, this is DNA, not protein, but proteins have NH2 groups. Okay, looking at this cytosine, that NH2 is chemically fragile. If you expose it to water, a chemical reaction happens. What happens is the NH2 pops off and an oxygen sticks there, and you oxidize it. This is rusting of the genome. This happens in your cells every day. You have about 1 million, they're called lesions. 1 million places in your genome in every single cell, every single day, where you either have a break or a chemical reaction in that DNA. And your repair enzymes are constantly scanning the genome and replacing damaged places. But here's the deal. That molecule on the right, that's not a random molecule. That's a uracil. The U in RNA, that's it. So when your enzymes are scrolling around, they find a U, say, what is U doing here? And they replace it sometimes with a thymine. So what we have is a C to T transition. The C to T mutation is the most common mutation in human history, and in bones in the ground. So when you sequence ancient DNA and you find there's a T there, everyone else in the world has a C, well, is that a mutation that happened in that person, or is it something that happened in the ground? And this is a massive problem. DNA is fragile. This molecule is notoriously easy to break. This happens constantly while we're alive. Think about it, after we die, all the repair enzymes stop working. Your DNA falls apart really, really quickly. So those three things mean bad news for ancient DNA. Okay, knowing that everything breaks down quickly, how do they get it? Well, this is what they do. They take a bone and they treat it like crime scene evidence. They wear masks, they wear a face shield, they're usually in a negative pressure room. They try to keep all the extraneous DNA away, and they don't touch it, because if you touch it with your fingers, an old bone, the DNases on your finger are going to destroy any DNA they run into, and your DNA, because there's so much of it, will overwhelm a little bit of ancient DNA that might be in the bone. So when was it Homo naledi? I have to remember this. this is a little aside. I shouldn't do asides because when I try to do an aside, I forget half the things until later on. Oh yeah, now I remember. But I think it was Homo naledi when they discovered it in a cave in South Africa. They had these really cute, skinny graduate students, females, go way deep in this cave. I mean, I don't know why there aren't any, ugly male graduate students who could have done it, but it was a nice publicity stunt. And they dug out all these bones from this cave in non-sterile conditions. And you ruined it. Because as soon as the human hand touches it, it's probably destroyed for scientific evidence as far as DNA is concerned. But they weren't thinking straight because we had already invented ancient DNA. They should have done it better because we might have been able to pull DNA out of those bones. Okay, here's what they do. They extract it in a sterile lab. And as soon as they get the DNA that's all fragmented, they tag it with a random piece of DNA. Random letters, 10 or 20 letters long. And that tag right there will be unique to each one of the DNA pieces. Then they do something called PCR. That's a polymerase chain reaction. They just take the little bit of DNA they have and they make lots and lots of copies of all those random pieces. And then they sequence all of it. 99% of that DNA will be bacterial because bacteria are living in the bone and they're eating the DNA. Another mystery. Why is there any DNA left after hundreds of thousands of years? I don't think it should be there, but they sequence it and then they have to authenticate it. And here's what they do. It's a two-step process. First of all, since DNA breaks down over time, you don't expect to find any long pieces of DNA. If you find a long piece of DNA that you sequence, it's bacterial or human, they just throw it away right away. It's gone. And then they look for a piece of DNA with lots of C to Ts and other mutations at the end of it. Because what they figured out chemically is the oxygen attacks the DNA from the ends and works its way in. So they look for a bunch of C to Ts compared to the human genome. You could not do ancient DNA sequencing without a comparative sequence. And then they cut the ends off. But there's an art to that, because if there's a C to T mutation in the middle, do you know that that happened in the person or did it happen in the ground? You can't tell. So statistically, there's a trimming algorithm, which means that in the end, we cannot be 100% certain about the sequence. So that's the secret of ancient DNA is it's good enough, but it's not perfect. And then they take all these little teeny. Now, when they sequenced the first Neanderthal genome, the largest piece they found was 50 letters long. Wait a minute. Our chromosome 1 is 250 million letters long. It's literally that long. It's like 6 inches long. And the longest piece of Neanderthal DNA they found was like 50 letters. And the ends are all mutated. So after they truncated the end, we're talking about very little pieces of DNA. You cannot use them to assemble a genome. But if you have a human genome, you can take those pieces and 20 letters. There's not many places in the genome that can have the exact same 20-letter sequence. So they just scan through the human genome. Oh, it goes right there. Take another piece. Oh, it goes right here. And they boop, 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 and they assemble it like a giant jigsaw puzzle. It's really, really cool how they do that. If it doesn't assemble well, it's not human. If it assembles very well, it is human. If we look at the decay rate of DNA, and we can measure this in the laboratory, it's shocking how fast this decays. By the way, this is from Brian Thomas and Jeff Tompkins, two good friends of mine. Very interesting work. Sorry, this is a reference to them referencing another study. Depending on temperature and depending upon the length of your DNA fragment, the half-life of the 30 base pair fragment at above freezing is about 3,000 years. If you can freeze it, it can last a long time. The problem is there's no place where we're finding DNA that's the temperature is always below freezing because people don't live in those places. Even in the Arctic, it gets above freezing. Now the ground might be frozen, a foot or two down in the permafrost, but the upper soil melts every summer. This is bad. The temperature, a freeze-thaw cycle is really, really bad for DNA. Either keep it frozen or keep it cool, but you don't want ice crystals to form and melt and crystals to melt and crystals to melt. That'll destroy DNA faster than anything. But looking at the half-life, the time to complete decay of a piece of DNA is way too short But complete decay is not even what we have to worry about, because you can't sequence individual letters. Now sequencing machines work. You need a piece of DNA. If you don't have 20 or 30 letters, you got nothing. And yet here we're finding in ancient bones pieces much longer than their half-life indicates, if they're as old as the evolutionists claim. I'm going to leave it like that. You can go to those references if you're interested, if you thought to write it quick. That's in the Journal of Creation. Type in Tompkins genome. I bet you'll find it. Oh, drum roll. More and more evidence is coming out of non-petrified bones in dinosaurs. This is one just from this year. Evidence of proteins, chromosomes, and chemical markers of DNA in exceptionally preserved dinosaur cartilage. Now notice they did not say DNA, but they said evidence of DNA. See, scientists are trained to never say something that you can be contradicted with. So they don't say, I found DNA. They say, the evidence supports, or the evidence lead us to conclude. That's a strong statement for scientists. Or the data suggest. Those are the phrases that we use. So here they're saying the data are suggesting that they're intact chromosomes. What? Wait. The dinosaurs supposedly died out 65 million years ago. Everything in chemistry, everything in biology, everything in the natural world is telling us that DNA isn't going to last tens of thousands, let alone tens of millions of years. This is powerful stuff for the creationist side of the argument. And they've got all these pictures, and they'll take a cell. I just said they take a cell from a dinosaur bone. How come that thing hasn't broken down? Anyway, they take a cell and they stain it for something that stains DNA. So on the far right, DAPI is something I've used in laboratory a lot. DAPI is a flat molecule that sticks into the DNA double helix. And then when you wash the cell with water, the DAPI stays there. It's fluorescent, so when you shine ultraviolet or purple or blue light on it, it glows blue or green. Ooh. But they can also use stains for things like histones. Now, a histone is a protein that the DNA wraps around. And one of the cool reasons for doing that is there's lots of bacterial contamination in these bones, but bacterial DNA isn't wrapped around histones. So if you find a histone in a dinosaur bone, you know you're dealing with intact cells, intact DNA. Oh, it's not supposed to be there, my friends, and yet there it is. This is following Mary Schweitzer's stuff from 8 or 9 years ago now, where she did the same thing with dinosaur DNA for this from the Hell Creek Formation in Montana. Sure enough, on the left, that's DAPI staining. On the right, that's histone staining. There's really good evidence of DNA in dinosaur bones. Now, has anyone looked to sequence the DNA? Yes, but remember, this is really old stuff. I mean, Neanderthals are post-flood. This is flood. This is hundreds, if not 1,000 years older material. I really expect it to be decayed, but there's enough there for the stain to work. As DNA science advances, I predict we're going to be able to sequence DNA from dinosaurs. Does that mean there's going to be a Jurassic Park? No, because the DNA is highly fragmented. You cannot take this and stick it into an organism and have it live. But we might be able to tweak DNA of a reptile and make it more dinosaur-like. In fact, I wrote an article a while back in Creation Magazine, not Neanderthals, mammoths coming to a zoo near you, question mark. I was talking about the efforts of scientists to clone a mammoth. And the answer is they're not going to clone a mammoth. But we've sequenced the mammoth genome. We can theoretically genetically engineer an elephant egg. Oh, this gene gives them hair, change the elephant to make it hairy. This gene gives them the big tusks, change that gene to give it the big tusks. And we might be able to recreate a mammoth from an elephant, which is really cool, but technologically challenging because elephants aren't lab rats. Much harder to work on an elephant, but it might happen someday. Okay, let's do some case studies now. I said earlier we're going to go backwards. Let's look at Native Americans first. First, when we look at the way Native Americans look and we look at their languages, they're very similar to people living in like the Kamchatka Peninsula and Northeast Asia, some of the Siberian tribes. But when we look at their DNA, clearly they are related to Northeast Asians. In fact, when we look at ancient DNA in the Americas, clearly the Native Americans are related to the most ancient people that we've ever found. And there doesn't seem to be any old African DNA or old European DNA in Native Americans. So that means when you see like an Olmec skull, sorry, not a skull, but an Olmec rock in Central America, the Olmecs, they carve these giant volcanic rocks and they look almost African in their appearance, but there's no African DNA in the modern population there. So it doesn't look like they really were African. We talk about the Vikings getting over to the New World in Leif Erikson's time, 1000 AD. Well, they apparently didn't leave any DNA behind. And there's a tribe in South Carolina that claims that they're an older migration of people from Europe coming over, but there's actually not any good evidence for that. Everything is pointing toward a Siberian, Northeast Asian origin. Now, Kennewick man was a very interesting discovery and super controversial. If you come across a land bridge from Asia, you have two choices. You have to walk through Alaska, and hopefully there's an ice-free corridor during the Ice Age where you can come down on the east side of the Rockies into the Americas. Or you can just skip along what they call the kelp highway. Because if you stay along the coast, there's abalone, there's kelp, there are otters, there's fish, there's crabs, there's tons of nice seafood, and it goes all the way down to Baja California. That's a highway for people to migrate. But as you're coming down south, you get into Washington. When you get to the Columbia River, there's this giant gorge through the mountains, and it's a natural place for you to get to the interior of the continent. Kennewick, Washington is along the Columbia River on the east side of the mountains, the east side of the Olympics, the Cascades, I guess it is. I'm not sure what it's called there. And it doesn't look like a Native American skull. And so there's lots of arguing. Some people said that's Asian. Some people said it was European. And since Native Americans are very protective of their relics, they don't like people digging up their old graves. They said, give us our ancestor back. And the judge says, no, he looks European. You can't have him back. But finally, someone did some DNA on it, and it turns out the Kennewick man not only is Native American, but he's most closely related to the people living in that area today. So Kennewick man, the bones were given back to the Native American Tribal Council, and they buried it. So is that a loss to science? Those bones are now rotting in the ground. They will never be used again. Is that a loss? Actually, no, I think it's a win, because we got the genome out of it. And the Native Americans were given back someone that they consider special. I don't blame Native Americans for not trusting modern genetic scientists. I don't blame them at all. Maybe we'll talk more about that later. All right, let's skip over to China. East Asia, not Northeast Asia, Siberians, but down a little lower. We have China, Japan, Korea. These people are very closely related. Somebody found, they say, a 40,000-year-old modern human from a cave near Beijing. He's extremely similar to modern Chinese people. And it looks like he was there after the Native American split from the East Asian population. That's really cool. We can tell all sorts of very interesting things about history from looking at ancient DNA. But we know the Chinese people have been in place for a very long time. And that has affected their genetics. In fact, we can see it. I'll show you a picture in a couple of minutes where you can see it. Okay, let's go further west. Let's go to Europe. Most of you, before I started talking, maybe even now, you would say, if I said, who settled Europe? You might say the Japhethites. And I'll say, oh, really? How do you know that? We actually don't. In fact, if you look at the earliest Europeans, well, they're Neanderthals. After that, the Neanderthals were replaced by a group of people that when we look at their DNA, it looks like they had dark skin. And a lot of them had genes that, if found in modern people, they cause blue eyes. So dark-skinned, blue-eyed people. That's very strange. And in Europe, because a lot of archaeology has been done in Europe, we have all these different archaeological levels, and we can tell which one comes after the other. Now, I don't agree with the dating, but I do agree with the order. First you have Neanderthals, and then you have a group of people they call the hunter-gatherers. Now, what is a hunter-gatherer? Think of a guy living in the woods. He's got a log cabin, he's hunting, and he's, gathering mushrooms. That's a hunter-gatherer. It's not primitive. It's just a person foraging like even some modern people do today. Okay, hunter-gatherers come first. What's my next slide? Oh, this slide. Oh, people were just saying, let's do this. This is crazy. Someone's doing an archaeological study in southern Denmark, and they find that brown thing. Now, that's not fecal material. That actually is tar from a birch tree. Apparently, lots of people chew birch tar. I've never tried it. Looks kind of gross. But when they found it, they said, oh, this is human teeth marks in it. Let's sequence the DNA out of this thing. And they built the entire genome of a female with apparently dark skin and blue eyes. But they also found nut DNA, apparently hazelnuts, and they found duck DNA. So apparently this little girl or woman, we don't know how old she was eating duck and hazelnuts for dinner, and she chewed up a piece of birch tar and spit it out in the swamp. And now, a couple thousand years later, we find it and we sequence her genome. This is nuts and crazy, but think about this. We know who was living in Europe, and the people that were there are not like the modern Europeans. Modern Europeans hadn't arrived yet. So even up in the evolutionary story, non-Europeans, Neanderthals, non-Europeans, and then Western hunter-gatherers. That's like this girl we're just talking about. And then in the so-called Neolithic, that means new stone, the new stone age. This is when some farmers are coming up from Turkey. They're moving into the area and the DNA is changing. Oh, it's changing a lot. They call them the EEF, the early European farmers. First of all, the SLC24A5 gene, yeah, that's this gene. I have 2 identical copies of that gene, and it's the main driver, one of the main drivers of my skin color. It wasn't in Europe until this time frame. You've heard of Otzi, the Tyrolean Iceman they found on the border of Italy and Switzerland. As an ice patch melted back, some hikers found this dead guy who had been dead for several thousand years. And it was shocking because he had tattoos all over his body. He had waterproof boots that were stuffed with fur to make them warmer. He had arrows that had actually been 2 parts, 2 different species of wood joined together. So when he shot the arrow and stuck in something, he could at least retrieve the part with the feathers. He had a pouch with ashes in it. Apparently he was carrying around coals to start his next fire. He had another pouch with mushrooms in it. Strange, but those mushrooms are a remedy for intestinal parasites. And he had that species of intestinal parasites, yuck. And on. This man was a very intelligent person who descended from people that looked like they come out of Turkey. So here's what I'm saying. After Noah's flood, before the Tower of Babel, you had all these people mixing together. At Babel, people spread out. But then there was another wave of people that came later. See, farmers have more food. Farmers have more children. Farmers live an easier life. They tend to live better. And a larger population starts blossoming from the central area around the Babel area, and that population re-expands and spreads across the world and replaces the first people who got to where they were. Here's an example of that. Now, where are modern Europeans in all this? See, even Otzi and even the early Neolithic farmers, they don't have European DNA. If you're looking for Europeans, you have to go out to the plains, north of the Caspian and Black Seas. That's Asia. Those vast grasslands that the Mongols swept from Mongolia and they came all the way over into Europe. Yeah, those grasslands. Dig into a big grass mound. They call it a kurgan. And dig through there and you might find some bones and maybe a sword or maybe a horse. Some ancient Bronze Age warriors buried there. And if you sample his DNA, it's quite possible that he has very European-looking DNA. because the Europeans weren't in Europe until the Bronze Age. This is probably when the Indo-European languages came into Europe. It's involved with a brand new set of archaeology, the corded ware culture. In other words, they take a pot when the clay is still wet. They either wrap it or they imprint it with twine. It just makes some ridges on the pot. It's just a traditional style that they were using at that time. For a long time, the archaeologists were arguing, did a brand new people come in and wipe out the first people that were there? Or did these people that were already in Europe learn a new pottery style and it was just a trend and they changed archaeologically without changing genetically? And now we know they changed genetically. So did the Europeans coming in kill off everyone that lived there? No. They intermarried with them. First of all, they were a larger population, so a smaller population, if they completely merged, they just got overwhelmed. There was probably a lot of fighting, and there was some periods in ancient European history where people groups were living side by side for centuries that were more different than Europeans are today from Chinese people. And yet these different groups living in Europe merged to become what we call European. So I get to ask the question again, what is a race? Hey, Europeans, that's your ancient homeland. It's not Ireland. It's not Germany. It's the steppes of Asia. That's cool. Anyway, as the Yamnai culture spread, let's take some time now and look at a Y chromosome family tree. Now, I made this. This took me forever. I had to download a whole bunch of Y chromosomes from the Simon's Genome Diversity Project. It's hosted in a lab in Europe. It took a long time. This took me days of work to make this tree. This is an evolutionary tree. I'm perfectly happy with this because of what it shows us. Now, I think you can see my mouse here. Looking over here, see this O? These are people that live in China, Japan, Korea. Do you see this fan shape? This beautiful wedge? This is people that started from a single male ancestor and have increased their population size consistently for a very long period of time. Small populations produce leggy things like over here. These are the Khoisan Bushmen of the Kalahari Desert, the pygmies that live in the African central rainforests. Very leggy, small populations. Over here, this E1B1A, this is 95% of Africans. 95%, but look, they have a recent common ancestor. And E1B1B, some of these are Africans and some Iranian, Palestinian. And E2D and C, Genghis Khan was a C. So what we see is 95% of Africans branch off from a non-African that branches off from non-Africans that branches off from non-Africans. Where do these people arise? They arose outside of Africa and then invaded and took over. Oh, my next one, the broomstick. No, okay, I'll stick with this for a minute. Down over here, R1B. I'm an R1B. Remember I was talking about that earlier? Look at our branches. They all come together pretty recently. In fact, Q, these are Native Americans. So as the Native Americans are coming across the land bridge, after that is when R1B starts originating in Central Asia. You just picture here. Does this look like an explosion to you? That explosion is the pattern we predict from a rapidly expanding population if it starts from a small number of people. This is exactly the pattern we'd expect in an Adam, Noah, a Tower of Babel sort of scenario. Now, if you rewound the clock and did it again, you get a different picture every time. But the general pattern of this spreading out from a central area, this is a signal we expect from the biblical story of the origin of mankind. And we can also read a lot of different history here. I just told you about China, Europe, Native Americans, and Africans. There's stories of each one of these branches that are fascinating. I'm going to read you something from two pretty famous guys, Slatkin and Racimo. Can I move my little thingy here? I can't. Oh, back. I gotta move my people's faces out of the way. They say this: we now know that present-day populations were created by a complex history of admixture and population movement. So even though the Table of Nations was written at a specific point in time, we've had 4,000 years of history since then. Although local genetic continuity over long periods has been documented in a few cases, these are exceptional. The general rule is that the ancestors of present-day populations lived somewhere else. When we look at ancient history, humanity has been fluid and moving. And that is really cool and really amazing. I talked about those 95% of Africans. Looking at Africa today, the people in Southwest Africa, the Khoisan Bushmen and live in the Kalahari, they don't have really dark skin. They have the skin color about Barack Obama. His mother was European. So they're not super dark people. They have the same genes for skin color as we find in Northern Africa and in the rest of the Middle East. Oh, that's interesting. In fact, the same genes for skin color in Africans are found in southern India, in Melanesia, and all across Australia and Tasmania. The same exact genetic variants. So if you might be thinking that Ham was cursed with dark skin because Noah cursed him, you are wrong. First of all, the Bible doesn't say that. It says Canaan was cursed, and Canaan lived up here in what we now call Israel. But second of all, the same exact genes appear in other places also. These genes are in the population before we spread out. These are Babel genes. These are Ark genes, probably Adam genes. But the same is true of the light skin color variants. They're found all over the world. The same, in fact, these light skin variants in Europe are found all over Africa too. It's just because some genes are dominant over others. If you're carrying a light skin variant, it doesn't matter. But those genes are there. Now the Bantu people, the most famous Bantus are the Zulus. And the Zulus live right down here in southern Africa, South Africa. When the Dutch got here, they found 100-mile wide swath of grassland that was just open. And they said, free land. No, the Zulus had killed off everyone within 100 miles of their homeland. And the Dutch walked right into that. And then the English inherited it, and they fought 2 wars with the Zulus. The Zulus are a Bantu people. The Bantu peoples originated in Central Africa. And about maybe 1,500 years ago, they swept eastward and southward, going across Africa, and according to the ancient DNA, killing off the people that originally lived there. Because when we look at the Khoisan people, the ancient DNA of the people in the rest of Africa looks very Khoisan, and it doesn't look Bantu. But the Bantus are now living there. Now, I just said the same thing about Europe, so I'm not focusing on any one people group. In fact, the only reason you're alive right now is because your ancestors killed off other people. You are the product of thousands of years of warfare. Human history is really bloody, so I'm not singling anybody out. But looking at that, this means that we have evidence of an expansion, we have a genetic change, we tell all these really cool things about history from all these neat genes. And I get to ask the question again. If the people in southernmost Africa had the same skin color genes as the Iranians, or if the people in Central Africa have the same skin color genes as Indians, what's a race? It's not based on skin color. Okay, last little section, Neanderthals. First, a long time ago, they found Neanderthal mitochondrial DNA. It's clearly human, but none of those variants are found in any mitochondrial DNAs in modern humans. They finally sequenced a partial Y chromosome a couple of years ago. We got a lot of female Neanderthal skeletons, but only one male. That's really odd, but they did partially assemble a Y chromosome, and it's very different from modern humans also. Okay, this is true. But we now know that modern people living outside of Africa, see, Neanderthals didn't live in Africa. So even though they picked up Neanderthal DNA in Africa, it's only little pieces, and probably because people from outside of Africa came into Africa. But everyone else in the world has about 3% or so of Neanderthal DNA. Now that doesn't sound like a lot if you put it in this context. For every one of my ancestors, 3 of them were Neanderthal. Or put it in this context. If I've got 50% of my parents' DNA, 25% of my grandparents, 12.5% of my great-grandparents, 6% of my great-great-grandparents, 3% of my great-great-great-grandparents. That's the same amount of Neanderthal DNA that I carry. There's a lot of Neanderthal DNA in modern man. Meaning, one, they're Homo sapiens. Because we interbred with them, we carry their DNA. Two, when we look at their genetic variants, they carried some skin color genes that might have given them light hair, light eyes, freckles. Oh, wait a minute. That's really cool. But we know they interbred with humans on more than one occasion. We have found individuals that had a Neanderthal, 5 or 6 generations ago. And earlier people have bigger chunks of Neanderthal DNA than modern people do, because over time it's gotten scrambled and broken into little smaller pieces. We found, okay, I'm going to skip to another group of people, the Denisovans. They come out of a cave named after a monk who used to live in this cave. Now this is way east in Siberia. This is close to Kazakhstan and Western China and Mongolia. This is really far east in Siberia. They've been digging in this cave for a long time, and this is where the first Neanderthal, this is where some of the significant Neanderthal genomes have been built from bones in this cave. But then they found a finger bone. Actually, I think it was the left hand, the end of a pinky, and they found DNA in it. But it wasn't Neanderthal, and it wasn't modern man. It's a brand new group of people. Almost as, if you look at like the lines, they branch off from Neanderthals almost as soon as Neanderthals branch off from modern humans. So they're slightly closer to Neanderthals than modern humans, but not by much. They call them the Denisovans because they come from Denisova cave in Russia. Well, it turns out that about 0.5% of Asians are Denisovan, but Europeans don't carry Denisovan DNA. And when we look at the DNA of Melanesians, up to 7% of them are Denisovan DNA. Whoa, but they're also 3% Neanderthal. So they're up to 10% non-modern. Ooh, that's scary. Because if they're up to 10% caveman, think about what you can conclude from that. And yet, when you look at these people, they're perfectly intelligent. They're perfectly smart. They're industrious. They are, in fact, normal, everyday human beings. Because the Denisovans are also human, like the Neanderthals. It looks like the Tibetan population got a specific piece of DNA that helps them tolerate very high altitudes without getting altitude sick from the Denisovans. And in fact, they found in this cave a female skeleton, another female for some reason, but a female skeleton who was half Neanderthal and half Denisovan. Now, we know that these things interbred over time, right? What's the probability over tens or hundreds of thousands of years of events happening? And we see today we have their DNA, but what do you think the probability is you're going to find that individual? It's going to be incredibly remote. The fact that they found that individual tells me that there has not been tens or hundreds of thousands of years. These things happen quickly. And a lot of the bones we're finding them, well, that's a significant fraction of the population we've already dug up. But when we look at this woman, this girl at least, the Denisovan part, okay, that's Denisovan, but the Neanderthal part was not related to a Neanderthal skeleton buried 3 feet away. The Neanderthal part was related to some Neanderthals they found in Croatia. Wait a second. If you started walking in Croatia and you started going east, you would never find Denisova cave. It would be literally impossible to just randomly stumble upon it. The fact that we found genetic continuity between these two places means that Neanderthals, even though they had an incredibly, at most there's only a few thousands of them at any one time. Maybe 10, maybe 50,000 at the top. There was never hundreds of thousands of Neanderthals, but they knew where each other were. The fact that the ones in Croatia, similar DNA can be found all the way east in Asia means that there was a trading network among them, an exchange of genes among them. This is really interesting. All right, I'm going to wrap up here. Takeaway points for you. 1, ancient DNA exists. It should not. If it exists, Bones are not as old as the evolutionists say. 2, we can learn a lot of really cool things about ancient history from ancient DNA. I just threw a whole bunch of things at you. 3rd, it does not contradict the Bible, but we have to get our biblical thinking on straight. I don't think the Table of Nations is describing every people group in the world. I think it's describing all the people groups within a stone's throw of Israel, or wherever that person was when they wrote it. The evolutionary time frame is directly contradicted by the presence of ancient DNA. Now, we have gotten a lot of curveballs, things we didn't quite expect, but you know what? This is a brand new field. Everyone has gotten curveballs. Nothing I just told you would anyone would have expected 15 years ago. This is brand new, fun, and amazing information that's not super challenging for the biblical model. Now, there's a lot of other subjects I did not cover. If you go to creation.com, you can get a lot more information on any subject you like, but even about DNA, and I've written some things about ancient DNA also. I would highly encourage you to take a look at Creation Magazine. Now, I can't get you to sign up here, but if you go to creation.com, it's easy to sign up for Creation Magazine, piece of cake. Now, I say that because we get more comments from Creation Magazine than we do from our website. And our website's creation.com. That's a pretty important piece of real estate on the internet. And yet, our magazine, I don't say get more comments. We know more people read our magazine than read our web articles based on the number of countries we send it to, the number of people we send it to, the average readership, and the comments we get back. Even though creation.com is really important, Creation Magazine actually is one of the most important things that we do. So if you don't get that magazine, we're going to really encourage you to take a look at it. Easy to find on creation.com. We can also sign you up for the digital version of the magazine. So if you don't want a paper version, you can read it on any device you have, and we'll let you share it with other people. We'll send you an email when it's ready. You click on the link and you can open up and read the magazine, really amazing stuff, and you can forward that on to whomever you like. Now, we say 5 people. We actually don't count. Don't tell my boss I said that. There's other things out there for you. If you haven't read the Creation Answers book, that's a first read. If you're basically just getting into this whole creation idea, this is going to answer about 99% of your questions. We do have some other amazing resources like Refuting Evolution 2, Refuting Compromise. We have a whole refuting series by Dr. Jonathan Sarfati. I've done several things on genetics. Here's one from a while ago. I did this at a Australian super conference, Creation Super Conference. Mitochondrial Eve and the 3 daughters of Noah. I'm claiming there's evidence of Adam and Eve, Noah's flood, and the Tower of Babel in the human genome, and the evolutionists have discovered it for us. And I lay out the evidence in that talk. This is probably my favorite talk I've ever given in my life. It's called The High-Tech Cell. What I'm trying to do here is explain how the human genome works, but no one understands how the human genome works because it's too complicated. So I compare it to a computer, and very quickly it gets into a hyper-complex computer because the genome is not simple. The more complex the genome is, the less able evolution is to explain it. If life were really, really simple, all right, evolution might be true, but life is not simple. It's the opposite of what evolution predicts, including the genome. Here's evolution's Achilles heels. This is a fascinating project. This is actually one of my favorite things I've ever done. This is 3 years of my life that went into this. What we did is we assembled 9 areas of science where we think the evolutionist thinks that it's a good argument. We said it's a terrible argument. And we asked experts in all those fields to identify places that evolution could not answer. And we call them evolution's Achilles heels. The book and the documentary, there for you on creation.com. I'm pretty sure you can get that that documentary. You can stream it or do a digital download. Here's Genetic Entropy and the Mystery of the Genome, the last resource I want to point out to you. This is written by my dear friend, Dr. John Sanford, who was an atheist for most of his life and then became a Christian about 10 years before he retired from Cornell. And about 5 years before he retires, he writes this book called Genetic Entropy and the Mystery of the Genome. And it horrified all his colleagues because here's an insider's insider explaining evolutionary theory to the rest of us. Now, he's an insider. I mean, you've probably heard of genetically modified organisms or GMOs. He invented the ability to make GMO plants, crops, his invention. So yeah, he is a big-time geneticist, a big-time evolutionist who came around to explain to us why evolution doesn't actually work. Amazing book that is. I'm going to leave you with 1 Peter 3:15. Very important verse. In your hearts, honor Christ the Lord as holy, always being prepared to make a defense to anyone who asks you for a reason for the hope that is in you. You do it with gentleness and respect. Now, I tried to show excitement for my topic, but I also tried to avoid polemical statements that would make people mad for no reason. I didn't insult anybody, I think. I tried not to be sarcastic, and I tried not to call anybody names. Why? Because that type of arguing, yeah, you might win a fan club, but you're not going to win any points with your opponent. It's actually a bad way to argue. And we don't have to do that. We can stand competently and confidently on the Word of God. We don't have to sacrifice the Bible for the sake of science, because science, thought about correctly, backs up the Bible.